Okay, so we will wait a few more minutes before we start. Hello, Peter.
Oh, hello. It's good to see you guys.
Yeah, good to see you.
Yeah. I was a little worried that maybe no Americans would show up because it's a holiday here, technically.
Oh, is it? Okay.
Yeah, yeah.
Yeah, yeah.
Oh, Michael Arit, that's good. That's cool.
Hello, Peter.
Oh, hello. It's good to see you guys.
Yeah, good to see you.
Yeah. I was a little worried that maybe no Americans would show up because it's a holiday here, technically.
Oh, is it? Okay.
Yeah, yeah.
Yeah, yeah.
Oh, Michael Arit.
Okay. Sounded like we had an echo of our conversation,
Mm-hmm.
but now it's gone. Susan, how are you?
I'm okay. California could be doing better, but you know, it was only 109 yesterday outside my house and smelled of smoke and whatever else. And we're only, unlike TJ, we're only at about 20-25% occupancy at Berkeley. So I have 4 people at a time in the lab. So I have 12 people a day and 3 shifts, but you know, they're not happy. They're getting work done, but it's not fun.
Oh, that's very unpleasant.
But it's going to be like that for a while. We've had no transmissions within the labs. So whatever we're doing is working.
Yeah, when we did these 2 shifts, Monday through Wednesday, and then Thursday, Saturday, I felt that maybe it's not fair for the second cohort that they have to work on Saturday. So we decided to shift, change every 4 weeks. But I felt that maybe we're not even going to change, right? It's not going to last 4 weeks before we move to the phase 2. But it looks like we're going to be staying in phase 1 for several more months.
Wow.
You know, lucky for us, we can get data and then work at home and process the data. I think it's a lot harder for the molecular biologists who need to be there often, but people are working on it. I think it feels like a ghost town when you go into the buildings. It's very quiet.
Oh.
Actually, it is much easier to keep safe distance when we do like microscopy, because you have one person in a small room. But in molecular biology, I just say if you need to work in the wet lab, it's actually much harder. So that became the most precious actually workspace in the lab.
Yeah.
So we have a strange situation where everybody works in the lab, but on the other hand, we have a huge second wave in Israel, and they are talking about the second closure here, and it's weird. Yeah. Okay, I guess we can wait perhaps one more moment and then we'll start.
Numbers are still growing.
Yes, the numbers are growing.
Ben, how is it in Switzerland, the situation, the policy?
It's relatively relaxed. So we are back in the lab, I would say to 70%, trying to keep occupancy low, but it's manageable. But we have a slow second wave starting. We're sort of increasing gradually. And so they're increasing the restrictions again. So we'll have to see how that works out.
Yeah. Okay, the 2 big deadlines will be November 3rd and then the winter. We'll see what happens in the world.
Yes.
Okay, so I guess we can start.
Yeah.
So hello everybody, welcome to the Protein Folding and Dynamics webinar. I think this is our 10th lecture, and we are very happy to have you today. Our lecturer today is Professor Taekjip Ha, who is the Bloomberg Distinguished Professor in the Department of Biophysics in Johns Hopkins University, and is also a Howard Hughes investigator. Taekjip also likes to be called TJ, started his career as a PhD student in Berkeley with Daniel Shemla and with Shimon Weiss, where he started working on single molecules and did some real pioneering work, like the first FRET experiment on the single molecule level and other experiments like that. He then moved on to be a postdoc with Professor Steve Chu at Stanford. And in 2000, he started his independent career in the University of Illinois at Urbana-Champaign, where he was for 15 years. And in 2015, he moved to Johns Hopkins. Taekjip is really one of the leaders and pioneers in the field of single molecule biophysics. And I don't think I will exaggerate if I will say that he is credited for being one of the people to actually making this field what it is today and making it absolutely important to do studies on the single molecule level when it comes to protein dynamics, when it comes to machines, interactions of machines with DNA and with other parts of the cell and so on and so forth. We are very glad to have Taekjip with us today. And just before I give him the microphone to start the talk, I'd just like to mention that I would like to ask all of you to keep your microphones on mute. And at the end of the talk, if you have questions, please include them in the chat. And I will ask each of you to turn on your microphone and ask the question. We hope to have a very lively discussion at the end. That's what we usually have in these webinars. So we hope to have that today as well. So with that, I will stop my share and I will let TJ start his talk. The title of the talk is Conformational Control from Single Molecules to Biotechnologies. So please, TJ.
Gilad, thank you very much for the kind introduction and also including me in this wonderful seminar series. So we do single molecule biophysics experiments and focusing on mechanistic understanding of how the proteins function. And I'll be telling you about 2 examples and how we can use the knowledge we generate to design new biotechnological applications. And this is done through controls of protein conformations. So we know that proteins can have multiple conformations. So this is a paper by Bob Austin. I believe he was a graduate student of Hans Frankfelder in Illinois, Urbana-Champaign. So here myoglobin can have different substates, conformational substates. You can actually choose between them, for example, by changing the temperature. So here in our project, we like to understand the functional roles of these conformational substates and then use the information and control the conformations and develop interesting applications in biotechnology. So in this particular talk, we're not going to use temperature to control the conformation, but we'll be using light and also chemical cross-linking. There are 2 systems that we're discussing. They share the common property of unwinding the DNA. So here is a CRISPR-Cas9 protein that unwind the DNA into 2 strands in an ATP-independent manner. Is another system, ATP-dependent unwinding enzyme called helicase. And it is easy to understand why you need ATP to move along the DNA directionally to melt the DNA. But you can also melt the DNA without using external chemical energy, because you can use a binding energy between the protein and the DNA to melt the DNA locally, just a few base pairs in the seed region. And in this particular example, you can actually expand the melted region if you provide the enzyme with a guide RNA of a particular sequence that matches the DNA sequence. You can continue to melt the DNA base pair with the RNA strand, and Cas9 enzyme can actually sense that it has achieved a threshold base pairing level between the guide and the DNA strand, and then undergoes a conformational change to bring the nuclease domains to the correct locations for target DNA cleavage, which form the basis of genome editing. We know from structural studies there are multiple conformations of these enzymes. In fact, from the drawings, you can see green and red the dots. Our labs and many other labs have actually used FRET for fluorescence resonance energy transfer at the ensemble level and single molecule level to study conformational dynamics of these proteins during their function. So let me begin by introducing the first system, CRISPR-Cas9. So CRISPR-Cas9 is adopted from bacterial cells, but for largely mammalian genome editing. And Cas9 is shown in the back as the cloud. And it is essentially a pair of scissors. It cuts the DNA at two locations to create what's called a DSB, double strand break. But it is programmable because you can actually guide the Cas9 enzyme to anywhere you want in the genome by using RNA sequence that matches the DNA sequence. So it starts from recognizing a short patch of DNA sequence called PAM, just 3 or 4 nucleotides, and then melts the DNA locally close to PAM proximal region, and then base pairs that region to the guide RNA, and it can continue to melt the DNA and exchange the strand to form additional DNA-RNA hybrid duplex, eventually undergoing a conformational change to cut the 2 strands. So once you generate this DSB, then the cell will take it over using these existing DNA repair mechanisms to repair the DNA damage. In the process, creating an edited genome that you desire. However, these programmable seizures are not synchronous. They are highly asynchronous seizures because when you express or produce the Cas9 protein and the guide RNA inside a cell, it takes a while for them to be produced. And it takes up to an hour for an active Cas9 protein to bind the target sequence in the crowded mammalian nucleus. So actually, when you do experiments, you actually don't know exactly when the cleavage occurs, really making it difficult to understand the kinetics of DNA repair or genome editing. So our goal here was to do better. We already have high precision along the genomic coordinate in CRISPR-Cas9. Can you achieve high precision in time to achieve a seconds level resolution, and in space to achieve a micrometer length scale control of CRISPR activity? And this work was done by Yang Liu, a postdoc, and Roger Zhou, a grad student, in collaboration with my colleague, Professor Bin Nu at Johns Hopkins Medicine. So concept here is the following. Imagine a robot sitting on the DNA where you want in the genome, patiently waiting for your signal that you deliver using light. Then, only then it'll cut the DNA quickly. This can be a great way of controlling genome editing, and also a great way of studying DNA repair, because now you can synchronize the reaction just like in protein folding studies, you can synchronize a reaction by using light or chemical induction. So we call this technology very fast CRISPR on demand. Here we allow the Cas9 protein to bind the DNA, but not cut the DNA by installing a large blockade that can be photocleaved. And then we shine light to remove the blockade, letting the pair of scissors close and cut the DNA very quickly. How do you do this? We use now well appreciated features of CRISPR-Cas9 system. On the left side, we have Cas9 protein in the back carrying the guide RNA. Red sequence is the matching sequence for the genomic location. It binds a PAM sequence and melts the seed region using the binding energy and base pair the seed region to the guide RNA. And if the base pairing is good enough, you can continue to melt and undergo strand exchange to additional degrees. And it turns out if you have 17 base pairs of matching between the two strands, then you can induce a conformational change into a cleavage competent conformation to make two cuts. And then Cas9 dissociates, you can start the repair of DSB. But at least in vitro, it's been known that you don't need 17 base pairs for a very stable binding of the protein to the DNA. You only need about 10 base pairs of match starting from PAM. So there's a disparity that we can actually utilize. So what it decided to do was to install photocleavable caging groups on the PAM distal side of the guide RNA, so that you still allow very stable binding of the Cas9 to the DNA, but not cleavage because you cannot have full DNA unwinding. Then you shine light after Cas9 has been pre-bound to the DNA to remove the blockade. Then DNA unwinding will occur quickly and activating the cleavage domains and getting the DNA breaks made. So if you want, you have all the cells basically getting ready to start, Cas9 proteins in the bound state, waiting for the signal that we deliver using light. And then, only then, we have synchronized cleavage that can start the reaction. So this works actually really, really well, even inside the living cell. So this is the primary data I can share with you today. So our method, very fast CRISPR, is shown in the red curve. Y-axis is the DSB fraction, basically what fraction of DNA inside a cell is cut at that time point. Even at the earliest time point of 30 seconds after light, we see more than 50% of DNA has been cleaved. So that's really, really fast, even compared to the previous best shown here using chemically inducible Cas9, which took about an hour to reach the maximum activity, which is still lower than our activity at 30 seconds. So our technology can cut the DNA very fast within seconds after shining light. So this was really exciting. There are many ways to generate genomic DSBs using chemicals or radiations or site-specific endonucleases. But compared to them, very fast CRISPR is better in all respects. We create a highly well-defined clean break at a specific location, but the target sequence can be very generally picked because you can just change the guide RNA sequence. You're targeting any place in the genome. It's very fast, seconds. We have a very high efficiency. So to demonstrate the capability of obtaining new information, let me show you one example. So once you make the DNA cut in the genome, you start the DNA repair by bringing many different DNA repair proteins to the site of DNA damage and some kinases and so on. And one thing that I want to highlight here is called gamma H2AX. So H2AX is a special histone in the chromatin. And upon damage signaling, that histone becomes phosphorylated making this modified histone called gamma H2AX. We know from other studies that this modification can spread to mega base pair size regions surrounding the cut site, preparing the DNA for DNA repair. But because there was no way to synchronize the DNA damage at high time resolution, we didn't really know the kinetics of spread in cells. But with very fast CRISPR, we can do this if you use a technology called ChIP-seq. ChIP-seq stands for chromatin immunoprecipitation. You have chromatin fragments from the cell, and then you use an antibodies against a protein of interest, a modification of interest, to capture these fragments, then sequence and map it to the genome to see where on the genome the protein is bound, in this case as a function of time. So we shine light to cut the DNA. We harvest the cells at 2 minutes, 5 minutes, 15 minutes, and 30 minutes, and perform ChIP-seq in a time-reserved manner. And then we can measure the kinetics. For example, in this case, the kinetics of gamma H2AX spreading, this histone phosphorylation. So this is the kind of data we get from ChIP-seq experiments. At the beginning, no light, we see just the background level activity, 11 megabase pair regions around the cut site. At 2 minutes, we see a little blip here at the cut site, which grows in height, and also it expands in width. Width is actually expanding gradually with time. You can plot the width as a function of time, show that the spreading is occurring in a linear manner in time, and at a speed that is, to me, astoundingly high, 150 kilobase per minute. And this type of information about spreading kinetics could not have been obtained previously due to the lack of synchronization method in generating DNA damage at a defined location. So I have shown you an example of extending the precision in genome editing from 1D in genome coordinate to 2D by adding a time dimension when you want. But can you do this where we want in 3D in space? If you can do that, we'll be able to do CRISPR genome editing in 5D. So here's an example of a spatial control showing CRISPR controlling at single cell resolution. Here there are 3 cells, and each cell is showing 2 green spots. These are due to 2 sites that we are going to cut, and they are bound by GAP-Cas9 fusion protein. In fact, the site actually many copies of the same sequence. You can generate many DNA cuts at the same time. And then once you shine light to DNA damage induction, we have M-cherry, red fluorescent protein fused to a DNA repair protein called 53BP1. And it'll be accumulating at the site as a marker of DNA damage repair response. So this is a snapshot at t=0 showing green dots. At 20 minutes later time point, we see the center cell has yellow spots due to the red repair protein coming to the damaged site, but 2 other cells still show green spots. And later on, these 2 spots turn red because actually due to extensive DNA damage, you fragment the chromatin. Let me show you the movie here. There's an initial laser dithering, and then these 2 green spots turn yellow and later a red. And in fact, the red cloud is larger than the green spot because, again, chromatin is modified in the megabase length scale extensively. And that's why we see larger spots. And 2 other cells do not show any change. They still show green dots, showing that we do have a single cell resolution control of CRISPR activity, which was really exciting for us. But we can do better. We can actually become more ambitious. There's nothing that stops us from focusing our laser light to the size of diffraction limit. So we can actually bring the laser light only to one of the two green spots and edit, cut the DNA at the single allele resolution. And this was actually a bit challenging. Took a couple of months for Yang Lu to make it work. So when it worked, he was so happy that he took out his iPhone, took a video and sent it to me. So I'm going to show you the first movie because that's fun. So yeah, there are two green spots and he zaps one of the two spots with the laser very quickly. You cannot even see dithering here anymore. And then that spot quickly collects the red dust. And this has been done now 100 times to build statistics and show that it's highly dependent on which allele you excite. So in the simulated allele, you see red cloud building up, but not on the other. And specificity was better than 80%, showing that we do have subcellular control and single allele resolution in potentially genome editing. So we think that there are many other applications that we can pursue in the future. And we think that with the subcellular activation, we can achieve high efficiency editing with no off-target effects. And editing a single copy of the two genes, two copies of a single gene could also have really interesting applications. And finally, there are really a lot of really exciting DNA repair studies that are enabled by this technology that we're also pursuing. So this part was done by Roger, shown here, and Yang, who was too sick to join our fishing trip. And our lab members are very happy here, having caught 2 fish each at the retreat in the Chesapeake Bay. Second part is another DNA unwinding enzyme, but that is ATP dependent. So these are helicases. Before I introduce helicases to you, let me just have one slide about a technology that many others and my lab also have developed. So this is the idea of combining single molecule FRET that actually I have this Gangnam style video that I have played on the normal circumstances. You use FRET to measure conformational changes of proteins, for example. But the other is optical tweezers, where you use a focused laser beam to grab a particle and apply a force to an enzyme sitting on the surface through DNA, for example. So our goal was to build a hybrid instrument to measure conformational changes using FRET or fluorescence, but as a function of applied force. So I'll be showing you some data obtained this way. And so if you want, in the optical tweezers, you use your hands to manipulate things mechanically in a purely mechanical manner, maybe with your eyes closed. But in FRET, for example, you use eyes to make observations, but passively with your hands tied in the back. But if you have both in the same experiment, you can sample the best of the both worlds. So that's the idea. So helicases are DNA unwinding or unzipping enzymes. They move on the DNA as a motor protein. But if you give them a single strand of DNA, that can actually work as a motor protein, moving on the DNA in a directional manner using ATP consumption. And some moves in the 3' to 5' direction and others in opposite direction. The DNA strand has a polarity there. So we've been studying bacterial helicases, names shown here, RAB, UVLD, and PCLA, for now 20 years. And they are DNA helicases that move in the 3' to 5' direction. They are generally actually poor unwinding enzymes. We see very little, if any, unwinding activity from a single protein. And there are two known major conformations called open and closed that differ in the orientation of the cyan domain. And the one question was, which of the two is functional for DNA unwinding? closed or open. And if one is functional, why do you need to have two different conformations? Most likely is for regulation of the activities. But how is unwinding regulated? So we use FRET to distinguish between the two states, high FRET for closed and low FRET for open conformations by selective labeling of two cysteines engineered into the proteins. And then we put that enzyme into a hybrid instrument that measures DNA unwinding using optical tweezers at high resolution, and conformation using FRET using a confocal laser excitation. So this is a tabulation of experiments done this way. So x is the FRET efficiency, y-x is the velocity. So in some of the trajectories, we see high FRET value causing due to close conformation of the enzyme. And the velocity here is positive, meaning that DNA is getting unwound. But the other conformation with low FRET state, that's the open conformation, velocity is negative, meaning that enzyme is somehow going backward. So what's going on here is that the enzyme is 3' to 5' translocase. It'll move along that strand to unwind the DNA ahead. After unwinding maybe 10, 15 base pairs, it switches over to the open form. So it was closed form during unwinding, and switch over to the open form, and then switches strands to the opposite strand to move along the opposite strand, the 3' to 5' direction, and causing the DNA to rezip in its wake. And this can continue back and forth repetitively, effectively preventing the enzyme from really active. So you unwind a little bit, you turn back, essentially inactivating the enzyme, until you bring a partner protein, we think, to stabilize that close conformation, disabling the open conformation needed for turning back. And then you can just continue to move in the same direction and unwinding the DNA to a large extended degree when you need to. And that is probably how in vivo unwinding function is regulated, through protein-protein interactions to stabilize unwinding active conformation. We have some data to suggest that is indeed the case. But this gave us the idea that maybe if you use a chemical cross-linking to stabilize the closed form here, you said two cysteines, and then you use bifunctional cross-linkers to cross-link them only into the closed form, not the open form, then you will disable the turning back of the enzyme and turn that enzyme into a highly processive and active enzyme. So that is the idea, and we call this variant RepX or PCLX because these are the names of the helicases, X for cross-linking. So with Jan Schemler's lab, we put this enzyme into dual laser tweezers and put the enzyme onto B surface, and then it is following the DNA to unwind the DNA. Then distance between the two Bs should decrease during unwinding activity. Indeed, when you put this into the ATP chamber, we see a rapid decrease in distance between the two Bs due to processive DNA unwinding and over thousands of base pairs. So this is really quite a surprising result, because without cross-linking, a single enzyme barely unwinds anything, maybe 10 base pairs if you apply unzipping force to the DNA. So you can convert this enzyme into a superhelicase that is better than anything else we have seen in nature by stapling the enzyme into the active form, and based on our understanding of how enzyme function regulation works. So that's our superhelicase here. So since then, we've been pursuing several applications of our superhelicase. I'll show more data here on using it to mimic co-transcriptional RNA folding. And I will show, I will not actually show you any data, but we're also using it for single molecule nanopore sequencing and isosomal DNA amplification. In fact, we can now amplify coronavirus. We are doing that in the lab. And for genome imaging, I'll show you some data there. So in terms of RNA folding, we know that RNA can also have well-defined 3D structures. And we think that the folding must occur during its transcription, not after all of the RNA has been produced. But it is actually not so easy to study RNA folding during transcription. So most of the studies have been done where you have a full-length RNA, and then you add magnesium to induce folding or refolding to study the folding dynamics and kinetics. But the co-transcription folding may not be the same as refolding, vice versa. So imagine having a piece of RNA that can have two alternative hairpin structures, black against the blue, blue against the red. And they may have equal energies, half and half in equilibrium. But if you let the RNA to fold during transcription, the first hairpin can form before the red segment emerges from the polymerase. And then if the exchange kinetics between the two stages slow, then you may not even reach the other state before an important decision has been made in the function of the RNA. So that's why we want to study co-transcriptional folding. And there are actually many other labs having pioneered different approaches for doing this. Now, our goal is to mimic this process using our superhelicase. So how do you do this? Basically, we want to extrude an RNA from a hole in this, the direction of a transcription 5' to 3' direction of the RNA molecule. Here's the idea. This is RNA we want to study in the green, gray color. We can label it wherever we want using the green and red fluorophores, so we can measure RNA folding before, after transcription. But we are nearly to a black strand, which is DNA strand that has a 3' tail. So our superhelicase, being a 3' to 5' helicase, will bind and translocate on the bottom strand to unwind the RNA DNA duplex and liberating the RNA in the 5' to 3' direction. And that's the direction of transcription. So we can call this vectorial folding. And it mimics a really probably most generalizable aspect of co-transcription folding, directional way of RNA production. It has the advantage of being able to put probes anywhere we want. So if the speed is actually also mimicking the speed of transcription, then it can be a really good way of mimicking co-transcriptional RNA folding. And because RNA, this helicase is very processive, you can do this on a fairly long piece of RNA. So does it work? So we use a very small RNA shown here, about 60 nucleotides in length as a test. So you can actually make the RNA, label it so that you can distinguish between folded and unfolded conformations using FRET. And then we add the protein and then add ATP to start the reaction. So in this case, we start with the RNA DNA duplex, and where you should have low FRET due to large separation. If our superhelicase unwinds the duplex it'll give you these two additional populations. And that's what you see. This is before we add ATP. We see FRET distribution at a low value due to the large separation of the duplex between the RNA and DNA. And 10 minutes after adding ATP, we see two additional populations, high FRET due to the folded state and low FRET, but not that low as in duplex due to the unfolded population of the liberated RNA. So that was good. We can also measure the kinetics of folding and unfolding after 10 minutes elapsed, and show that vectorial folding kinetics is the same as refolding kinetics you measure at equilibrium. So this was a good news, and we were excited that it worked at all. So we had beer. Then we got more ambitious. Can you also measure this in real time? The first time ever the RNA folds is allowed to fold, the maiden voyage. So for that, we can add our superhelicase to the immobilized DNA RNA complex and wash away free proteins. So you don't have to worry about additional proteins binding to the complex, and add ATP. As you unwind first 24 base pairs or so, we actually see an effect called PIFE, for protein-induced fluorescence enhancement, because protein approaches the green dye in high proximity, increasing viscosity and making the dye more rigid and higher fluorescence signal. As you unwind deeper into the duplex, then the liberated RNA coils up, giving you a transient increase in fret. Later on, it falls into a secondary structure in the and giving you a low fret of the undocked, unfolded structure. And later on, it also docks or falls into a tertiary structure, and you can go back and forth. In fact, we can see all of these features in real time. So initially, the green color shows increase and decrease due to this PIFE effect. And then later on, you see a blip, an increase in fret efficiency due to the collapse of the liberated RNA. And then secondary structure formation shown here, reduction in fret, and then tertiary structure formation, and then docking and docking going back and forth. So we are very excited that we can see this. And in addition, you can actually measure the time delay between different events and then deduce the speed of unwinding, which turns out to be between 20 and 40 base pairs. And this matches the transcription speed of bacterial RNA polymerases, suggesting that this can also be a good mimic in that respect. We have done some additional studies. I'm not going to show you the data here, but the paper is coming out this week together with Adrian's lab in NIH. So here we have a ribose switch that has two different forms, and in the off form, this termination hairpin form, so RNA polymerase will stop here and fall off. But the ligand binds to this RNA structure to form this aptamer. Then the competition occurs to actually remove the hairpin. Then gene is then turned on. So the outcome here then is a function of how quickly the RNA folding occurs, and how stable the folded structure is, and how quickly transcription occurs, and so on. So we can actually change the outcome by changing the energies of different states and speed of transcription and pausing at different points. It turns out we can use our superhelicase-based approach to mimic all of these elements of co-transcription folding, and really studying the effect of each effect at here without potential complications coming from other aspects of transcription machineries. So that was, theme of co-transcriptional RNA folding studies. We're also using our superhelicase for single molecule-based nanopore sequencing, and also amplifying the DNA at a constant temperature without having to use PCR, because if the helicase is doing the unwinding for you, you don't need to go up to 90 degrees to melt the DNA for loading the primers. Next, I will tell you briefly about another application that we are pursuing on genome imaging. So in this genome imaging field, we use typically DNA fish, and that fish stands for fluorescence in situ hybridization. So what you do is that you do global denaturing of the chromatin, DNA, and then you add labeled oligonucleotides that can bind to the melted DNA. And if you have enough of the dyes, you can see these genomic locations in a fixed cell. But because of global denaturing, your probes can bind to other sequences, and it can also compromise ultrastructures and imaging resolution and sensitivity. So my student Yambo Wang had an idea of what we call a goldfish. So this is basically using local denaturing approach. We call it genome oligopain via local denaturing fish. And here we do local denaturing by a superhelicase loaded at a Cas9 generated at DNA NIC. And we think that we can improve resolution and signal to noise ratio and fine structure preservation. So schematics is shown here. If you have Cas9 to bind the target DNA and make one cut using a Cas9 mutant that cut only one strand, then our superhelicase, RepX, can be loaded to the 3' overhang and translocase on the DNA strand in the 3' to 5' direction, causing the DNA to melt. But this can be done only locally. Then you can then bring in label probes to decorate only that region of genomic interest. So this works. And let me show you the image here. There are now 5 cells, and all of the 5 cells show this the cyan, the magenta spots. These are Sci5 labeled probes decorating 2 genomic sites that we cut and performed goldfish on. And in fact, I'm really impressed how robust this is. Essentially, most of the cells show 2 spots. We don't miss spots. I mean, there are some cells probably dividing. You can see up to 4 spots. In addition, we have really nice high signal to background ratio. In this case, 10 to 1. And this it comes from the fact that our specificity comes from DNA cleavage, not DNA binding. I told you earlier that Cas9 DNA cleavage has a much more stringent requirement for sequence match compared to DNA binding. So if you actually compare our approach, goldfish, in terms of signal to background ratio in the log scale to approaches that are based on just DNA binding, not cleavage, we see much better signal to background ratio due to the higher specificity. And that actually makes it possible to use this to measure genome conformations in high throughput and also even in patient tissues, that the application that we are pursuing presently. So I have just summarized different applications that we are pursuing that all depend on conformational control of a helicase based on our highly biophysical mechanistic understanding of their functional regulation. So one example that I showed you data on was to mimic co-transcriptional RNA folding, and the other was genome imaging using goldfish. So what's around the corner? Well, I think it would be really cool to control helicase function in live cells on demand. But the chemical cross-linking that we use is not really usable for live cell applications. So instead, we like to use light to switch between active and inactive state. And we are exploring several different approaches. One of them is actually what's called a LOB2 domain derived from a plant protein. And so basically, you insert this LOB2 motif onto the loop region of a protein, and then when you shine light, then this domain helix unfolds. So you start tugging on the loop and causing protein conformational change, and hopefully affecting its function. And this has been used by other labs to control kinase activities. So, we inserted the LOPT domain into our bacterial helicase in different positions. And here's some interesting data, still very early. So we have this flat-based ensemble assay. So if helicase unwinds the DNA, strand separate, the thread should go down. So if you just add this helicase with ATP, thread goes down due to unwinding. But if you then turn on the light, a blue light, then thread recovers. And if you turn off the light, the thread goes down again, suggesting that in this case, actually, although we kind of designed it to be light activated, it turns out to be light inhibited helicase. But we are still impressed because the fact that light does anything at all was impressive to us when you just kind of, inserted this LOPT domain is in some, location that based on our very unsophisticated guesses. So let me just acknowledge the people who have done the work. Sinan was our Superhelicase hero, and this was done with Jan Shemna's lab in Urbana-Champaign. And Matt was a postdoc who worked jointly between the two labs, and he is now in the faculty of Michigan State Physics, and a long-time collaboration with Tim Lohman in Washington University. For co-transcription folding approach, we worked with Sarah Wilson in Johns Hopkins and Adrian Ferrer-de-Marais at NIH, and they are wonderful postdocs. All right, so let me stop here by showing the slide. So this is our motivation in multiple conformations, understand them and find a way to control distributions between them. And two examples, CRISPR-Cas9 and helicases. And the goldfish is an application that actually combines both how our understanding of Cas9 specificity and also the superhelicase we engineered based on highly mechanistic studies. Well, thank you very much for your attention, and I'll be happy to answer questions.
Okay, thank you, TJ, for this really inspiring talk. And the floor is now open for questions. The first question is from Ninke Decker. So Ninke, please unmute yourself and ask the question.
Hi, TJ. That was very nice to see you talk or to hear you talk.
Hey, Ninke, how are you?
I'm doing good. I had a question about the superhelicase because it seems that you cross-link because you added to these cells, but then it seems to me that, wow, it could just keep going on forever. So would that not be a risk?
Yeah, so that's a really good point. First of all, the goldfish application is done on fixed cells. So, we don't have to worry about perturbing cells function.
Yep.
But if the superhelicase unwinds entire chromosome, then we lose the benefit of local denaturing, right?
Yeah.
So we have measured this actually, the length dependence. It looks like if you are more than like 3,000 base pairs away, we no longer see unwinding. So must be that there are proteins bound to the DNA that restricts the range of the superhelicase. So in that case, actually, it's nice because we melt DNA locally, right? And it's not a limitation. I think it'd be really interesting to produce light-controlled superhelicase in cells or chemically crossing superhelicase, maybe inject them into the cell and see what happens to the cell if the helicases are allowed to run amok, right? So that's another answer to your question.
Okay, great, thanks.
Okay, Ben?
Thank you very much, TJ. It's really incredibly impressive what you've been able to get to work here.
Thank you.
I have a question regarding your super fast CRISPR. Do you have any idea about how fast the actual start of the process is? So that the first jump that you see in the dead time of your experiment, any ideas what's rate limiting? How fast is this initial phase?
Yeah, so for cell-based experiments, we use 30-second illumination. So that's the fastest we can measure there. But we have also done this in vitro because you can run a gel to measure DNA cleavage. Mm-hmm. And there the time scale was 5 seconds. Now, Jennifer Doudna's lab, pioneer in the CRISPR-Cas9 field, they have also done some kinetic experiments. And it turns out the wild type Cas9 with the regular guide RNA, the speed of cleavage is 1 second. Basically it takes 1 second. So we are not there at 1 second, but we're not very far from it, right? So we cannot really go, we cannot do much better than 5 seconds because it'll be hard to improve upon what nature is already doing.
Yeah, very impressive. Thank you, TJ.
Thank you.
Next question, Trishit Banerjee. 2 short questions, actually.
Thank you so much, Professor. It was really a great learning for me. I had just had 2 short questions. One is, what difference do you see, especially in the kinetics of the unwinding due to just PAM DNA interaction and the other with the PAM DNA and the guide RNA interaction? And the second I had regarding was that the ATP you spoke about, that other than directionality, so how does it affect the unzipping in the DNA by helicases? And how does this unzipping compare to the unwinding process in Cas9?
Yeah, so so the first question is what happens if you just have Cas9 without the guide RNA? So we find that Cas9 binds the DNA, actually, it stays there for about 30 milliseconds and dissociates. So it probably melts locally, and then there's nothing to stabilize the structure, no guide RNA, so it dissociates from the DNA instead of staying there for a long time. And that's good news, and that's probably why they work, because otherwise, if they get trapped in a wrong sequence that contains PAM but no matching sequence, they will not be very useful, right? Is there any difference between DNA unwinding when it's triggered by an ATPase versus just binding energy induced unwinding? So I would say the binding and induced unwinding is more like a switch, where you go switch between unwinding competent and incompetent conformation. So whereas ATP ATP, when you need to use many ATPs, it becomes directional. And as one, I guess evidence for that is that when you use helicases to study unwinding using single molecule FRET, we see gradual changes in FRET, right? But in Cas9 case, we see just abrupt change. We don't see any intermediate, showing that it really does that like a switch on a really fast time scale.
All right, thank you so much.
Okay, next question by Anna Wipiewska.
Hello, can you hear me?
Yes.
Right. Congratulations for the presentation. It was really a pleasure to listen to your talk. And thank you to Gilad for inviting me here. So I'm just a very, very beginner in optical tweezers. I have done some FRET studies, and I have a question related to the studies of RNA folding. So what was the instrumentation that you used when studying RNA folding with FRET? Was it the combination of FRET and optical tweezers in that case, or? Oh,
so in that case, it was the purely fluorescence based. So we use FRET as a readout. There's no mechanical force being applied. Okay. So how did you immobilize the hairpin? Because I saw there is a hairpin and the probe is in the hairpin, but the other probe is just standing next to it on the slide. So how this is in reality? So we immobilize the RNA to the surface, usually using a DNA handle. So we extend the RNA by maybe 20 nucleotides, and we anneal it to a complementary DNA strand that has a biotin for surface tethering through biotin streptavidin interaction. We also often attach a dye to that DNA strand so that we don't have to modify the RNA with another dye. RNA modification is more expensive and more difficult. So when possible, we put one of the probes on the DNA strand that is complementary to the RNA used for surface tethering. Thank you.
Okay, I have a question about your helicase and the two conformations.
Mhm.
So we've seen recently examples where ligand binding to a protein does not really stabilize one conformation out of two, but just changes the ratio between the two conformations, even while there is very fast exchange between them. Do you have an idea whether a similar situation exists there, or whether it's really either one conformation or the other?
I think this is really, really wonderful question. I think a lot of parallels can be made to membrane protein studies. You know, your transporters, proteins, and ion channels, and structural biology and ion single channel recordings. You know, suggested that you have this like very well, you know, 2 or 3 discrete states of clearly different activities. But when people begin to measure single molecules using fluorescence and so on, they see actually more things, right? So you see changes in conformation that you measure by fluorescence that are not coincident with activity change and so on. And I think that you're also referring to something similar in your own system. Now, what we have done with our helicase, we know in vivo partner that activates the helicase. So we measured helicase conformation using single molecule FRET, but after adding that partner protein. So if you look at the FRET histograms, you see a dramatic shift to the closed form, the active form. But if you look at single molecules, it's not always staying in the closed form. It actually is. It is still going back to the open form. So I think this is exactly what you said, right? So you don't go from off switch to off position to on position digitally. You are changing the equilibrium population between the two by adding a ligand here. And I think that is really interesting, and also now based on other kinds of data available now, quite reasonable. Yeah.
Okay, thank you. Next question is Hagen Hoffman.
Yeah, thank you so much, TJ. This was really a wonderful talk.
Thank you.
And my question is actually rather technical. I was wondering what specific group did you use in the fast CRISPR system to actually block the cutting and then later on to activate?
Yes.
Maybe you said it and maybe I missed it. I don't remember.
Okay, so so this is a group called NPOM and NPOM, and it is attached to a thymine. So we replace a uridine in RNA by a thymine, but that is an NPOM group. And if you shine light, you basically get a thymine back without any scar left. And this was actually developed by Alex Dieters at University of Pittsburgh in chemistry for DNA studies, I think more than 15 years ago. And so we use that now for guide RNA. Actually, at the same time, the Dieters lab, they also use NPOM to control Cas9 activity. And that very nice Angevante chemistry that came out, even in zebrafish. But the difference here is that they put NPOM all across the guide RNA uniformly, right? So in that case, without uncaging, Cas9 doesn't bind the DNA, right? Then you shine light, then Cas9 can start to bind, but that takes about an hour. So you don't gain actually speed by using the case RNA. But in our case, by putting NPOM at the POM distal region, you still allow binding. And then from the pre-bound state, we get a very rapid activation. So that's the main difference here. So our technology, yeah.
Thank you.
Thank you.
Kun-Shi. Kun?
Hello, Professor. I just think about if whether your method, the VF CRISPR, can be used to detect the cleavage process for CRISPR case 12, because it only has one cleavage site for two different DNA strands, the target DNA strand and the non-target DNA strand. Yeah.
Yeah, the question is, can we use the same approach for Cas12? There's another, enzyme that also cuts the DNA based on guide RNA sequence, and I think you are wondering, because it uses the same nuclease to cut both strands, maybe it's not going to work as well. We don't know. We haven't tried that yet. There's another reason why you might think that it's not going to work, because at least for the version of Cas12a we have studied and some other labs have studied, it doesn't have this disconnect between sequence requirements for binding and cleavage. They seem to be sharing the same threshold. So in that case, it's not going to work. But then there's another variant, according to some other lab studies, that seems to have that also disparity between requirements for binding and cleavage. So if you use that variant, it may also work. I think that'd be an interesting thing to try. It'll certainly help you expand the kind of size that we can cut, because they have different PAM sequences.
Yeah, thank you.
Okay, we have another question from Ben Schuler.
Yeah, I had another question regarding your elegant idea to use the love domains for switching.
Mm-hmm.
I was wondering what's known about the kinetics of conformational switching of the love domain, because that's sort of a prerequisite. If you want to switch another protein, right, you need to know how fast the love domain switches and how much that depends on the photon flux that you need to use in the experiment to trigger.
Yeah, so I don't know the numbers of here. And I have to look it up, but I showed you this flow meter data where unwinding is measured by Fred when you shine light. Actually, it seems to reverse right away, and I think the switching is pretty fast. At least the time scale of interest to us. Another interesting aspect is that you can also come back to the original state spontaneously without having to shine a different wavelength light. And how long it stays in the light on state in the dark is a number that can be changed by mutations. You can have actually a mutation that has an extended light on conformation or shortened light on conformation. And that shortened one can be useful if you want to rapidly turn things back and on and off. But the extended one is better if you want to shine light once and then keep it there for longer. So you can perhaps reduce the photon flux required for an application.
Thank you.
Okay, it seems like we have one last question from Catherine Gosh.
Hello, can you hear me?
Yes.
Yes, we can.
Professor, it was a very interesting talk. I just had a small question. We know that ATP hydrolysis is highly coordinated in subunits of unmodified helicases. So do we observe such similar ATP hydrolysis in the subunits of the superhelicase despite such a chemical modification? And also, is this ATP hydrolysis control the translocation and unwinding rates in superhelicase?
Yeah, so I think that's a really interesting question. So, the helicase uses ATP hydrolysis to power through DNA. And now there's mounting evidence suggesting that it's one ATP per base pair. So there's a tight coupling between the chemical energy consumption and physical movement on the DNA. We have no evidence that cross-linking changes that coupling. And in fact, if you can also measure translocation on single-strand DNA after cross-linking, and we see the same rate as uncross-linked enzyme. I mean, we cannot rule that out, but that doesn't seem to have changed things very much. So that's good news, because now we have a really nice readout of processive unwinding activity without changing the elementary steps and the kinetics. This can be a nice way of studying the helicase activities at a really high fine scale resolution, which one approach that we are pursuing actually using the nanopores, because nanopores have very high resolution. And because our Superhelicase can unwind long pieces of DNA processfully, it's really nice, nice match.
Okay, it seems like there are no further questions. So TJ, I would like to thank you again for this wonderful talk and for a really inspiring discussion after the talk with many, many questions. And I would like to also thank the audience for being with us today, and to mention that in 2 weeks, we have another talk, this time by Matthias Rief from Munich. And thank you very much, and hope to see you all in 2 weeks. Bye-bye.